So, very delighted to have Dr. Patrick Ellinor with us today. Patrick grew up in Cincinnati, Ohio, and then went to Stanford for both his MD and Ph.D. He did his Ph.D. with Dick Chandler, who know him. And from there went to Boston. And in Boston, he went back to be trained as a physician in cardiology and cardiac EP, both at the Brigham and then at Mass General. And now he directs the Arrhythmia services at Mass General, and is very well known for his work on the genetics of atrial fibrillation, which he will talk about today and teach us all about it. Thank you for coming.
Well, thanks very much, Norm, and thanks everyone for having me out today. It's been a lot of fun to catch up with folks today. And this should be very casual. So if you have questions as we're going, if something doesn't make sense, I'm more than happy to handle them. So my interest in atrial fibrillation, as Norm and others earlier, I originally did structure function of ion channels when I was in Dick's lab, and was always intrigued by human genetics. So when I finished my, when you're doing your doctoral work, you then go back and do all your medical training. You come back to the lab and you're like 7 or 8 years out of date. So everything that you were doing is completely gone. And I was always intrigued by genetics and ran across one family that had autosomal dominant AF and kind of mapped that family and then started collecting them. That's what started it all. We just made it up as we went along. So it's been a lot of fun over the last couple of years. So these are the folks that keep me off the streets. Grants from a number of different sources. And then, why do we care about atrial fibrillation? I don't think I have to give a long prelude to this crowd, but as you know, it's about 1% of the U.S. population, 33 million people worldwide. You have a 25% risk of developing AF if you're over 40. That's for people in my neck of the woods. Okay, that's what it is for the rest of my life. But it's also important to realize that it's a similar prevalence to breast cancer, prostate cancer, and schizophrenia. Now, I'd argue you'd rather have AFib than any of these diseases, but what we actually know about the underlying molecular mechanisms of those diseases is actually quite a bit more than we do for AF. There isn't there isn't a single dedicated R01 or RFA on atrial fibrillation at the NHLBI. Yet there are about a zillion on breast cancer at a given time. So, it's something that, because it affects an older population, and if you get it and you have a stroke, you usually aren't a very mobile voter. So it just doesn't garner the same type of political attention. Why we care about it clinically is it's a five-fold overall increased risk of stroke, similar risk of threefold increased risk of heart failure, a dramatic increase in the level of dementia, as well as a doubling of the risk of overall death. And then costs a lot, and the treatment options are limited. I run a clinical electrophysiology service, and I'm happy when we do a lot of AFib ablations. It's the most common thing that we do. But frankly, that's fairly barbaric. You're going into the heart, you're barbecuing it, and you're trying to treat AFib. And I would argue that still, despite a lot of data by many investigators, we still have pretty limited understanding of molecular mechanisms. And it was, if you go back 15 years ago when I was a fellow, really the basic question that we asked is, can we just use genetics to try and pick apart the underlying potential pathways, as well as also identify increasingly what we spend our time in is looking at these as potential therapeutic targets. And I think that would have been crazy a while back, but when you now think of something like PCSK9, it doesn't sound so crazy anymore. So PCSK9, you have autosomal dominant families with a deletion in the gene, or even in people found in entire populations with a dramatically lower LDL level when you have a mutation in PCSK9. We know that it's safe because people can live with a deletion of the gene, and they seem to have all their fingers and toes and mentate normally. It has a dramatic reduction in coronary disease, and then just 10 years after it was discovered, a therapeutic was made, and within 11 years it was on the market. So the ability to translate genetic data into therapeutics is really dramatically shorter than it ever was in the past. So what's our approach? Basically, what I probably I spend most of my time doing are large-scale genetic studies. The other pieces are, we try to pick apart the functional mechanism or the functional genomics, what are the regulatory elements that are going from a genetic association through to a causative gene. And then lastly, what I spend about half of my time on now is actually looking at whether these genetic targets are potential therapeutic targets. And may have sounded a bit crazy, but we're making progress. I'll show an example from each of these, not so much this one yet, this is just 2 years in, we have a lot of data there. To do the genetic studies, we're focused not on individual families. Although I do still work with families, I love to work on them, they're fun, they're like my hobby. They're about my contribution to the lab, but mainly what we've been doing is dealing with entire populations. And the way that we do this is, for atrial fibrillation, like any other trait or disease, there's a genetic consortium. We helped start the Atrial Fibrillation Genetic Consortium. This slide is woefully out of date. It has about 70 or so different studies throughout the world at this point, but a good description of all the things that have been published are at our consortium website. We meet every 2 weeks by teleconference. We've done that for 8 years. We meet in person at AHA or Heart Rhythm Society or sometimes some other meetings. And we have a great group. I mean, basically, if you have genetic data, you're willing to share it and you're nice, we love to work with you. So we have a no jerks rule, but increasingly that drives a lot of my science. And when you have genetic data, you have to collaborate in order to find anything. The idea you're going to find anything in a population based on your 100 or even 1,000 samples, you just can't do it. So the collaborations are obligatory. And just as a reminder for those who may not be familiar with it, what a genome-wide association study is, we're taking populations. So we're looking for, and you can do this for any disease, but we've got a group of people with AFib, we've got our controls without AFib, and my little red stick figures, we're looking for genetic variants that are either increased or decreased in frequency in your cases versus your controls. It doesn't matter whether it goes up or down, it just has to be different. And then from these individuals, we isolate genomically negative blood sample, you throw it on a robot, it'll isolate the DNA, and then you genetically fingerprint. This is one array. The technique doesn't matter so much, but what you get is a sampling of, these days, a few 100,000 to a few million genetic variants throughout your genome. So we're each made up of 3 billion building blocks of DNA. One of these arrays, if you're white, you only need about 350 to 400,000 genetic variants to capture 90% of the difference between any one of us. You need a bit more if you're of Asian or of African American descent. But what's shocking is that this thing even works. Right? When you think about it, you take this little sample, you can compare a lot of people here versus here, and to do these comparisons, it's kind of striking to find anything in my mind. But what we do is we take these million genetic variants from one individual. We can take 300,000, we'll infer the genetic variants for another 8 to 12 million. You can calculate that pretty accurately. And then we compare them. Now, when you're doing 1 million t-tests or 12 million t-tests, obviously you can't use a p-value of 0.05. So we then typically will just do a Bonferroni correction of 0.05 times the 8 to 12 million comparisons that we're going to do. And this is where we stand for the genetic data. This is a paper led by Ingrid Christofferson from our AF Genetics Consortium, and this brings you up to date in one slide about where we are with the published data for AF. So this is a so-called Manhattan plot, which is we've plotted the minus log 10 of the p-value, so increasing statistical significance here, plotted against chromosomal distance. And each little dot, each little thing like the size of the pointer, each little dot is one genetic variant that we've tested. And so you'd expect if you do 12 million tests, most of them are going to be in the noise. By default, most of them should have no association with your disease. And what you're looking for are genetic variants, like this one at PIDX2 that we'll come back to, that all are located next to each other and are all significantly associated with your disease. Now, in the case of PIDX2, this is 1 times 10 to the minus 150. So we're pretty comfortable with that association. We've hatcheted this just to make the others identifiable. And what you see here are some things that you can actually understand, some that we don't have a clue about. So TBX5, that's well known in development. You see in 4 is the funny current. So you can pick out a few of these. What's kind of striking is the other things, right? Most of it are not things that we understood. And the very first study that was done identified this gene PDX2, which at the time, in 2007, people didn't have a clue. There's no one out there, although it had been well studied from developmental biology because it's an asymmetry gene, there's no one that would have put AFib and PDX2 in the same sentence. And now it's the most striking finding. There's been a lot of downstream work on it. This came out in the springtime, but what we're in the midst of is a fairly large explosion of genetic data. So this paper took I called Ingrid kindly the email postdoc, because she spent 2.5 years collecting the data from all of the different sites, from these 29 different studies, quality controlling it, going back and forth. So what we've done since is to basically pull together a lot of data by bringing it to us. So we bring the samples to us, genotype them, and then, and that's led to a dramatic expansion. So for our GWAS studies, we have our AF Genetics Consortium. We brought together 21 other studies and work that was a collaboration between Broad and Bayer that I helped to lead. And we fingerprinted those, we genotyped the data, we gave it all back to the investigators, and we just said, we just want to work on AFib. You can have it for whatever else you want. We've also pulled together the UK Biobank Data. If you're not familiar with this resource, it's an incredible resource for doing genetic and or epidemiologic work. In the United States, as much as I love my colleagues in Framingham, and I've worked with, had great collaborations with them, the Framingham data is essentially behind a firewall. And it's very hard to say get into epi data. They'll tell you otherwise, but this is true not only in Framingham, but of other large epidemiologic studies. The UK Biobank took a different approach. 10 years ago they started. They had enrollment centers for 500,000 people throughout the United Kingdom, and they collected clinical, longitudinal medical health records and genetic data. They did this fingerprinting, and they put it all on the web. And they said, go crazy. And so any of you can write an application for $3,000 to help offset the cost of it. You can get the entire data set. And as long as you don't try to identify individuals or do anything that would be unethical, you have as much of a free shot at it as I do or anybody in that program. So it's a great resource if you're interested in exploratory type analysis. We brought together a few other studies. We're in the midst of also genotyping. A buddy of mine runs the TIMI studies, so we're genotyping a large number, including the ENGAGE study. So when you put all this stuff together, we'll have more than 100,000 cases with AF for this fingerprinting. And to complement that, that gives you a fairly shallow, we're capturing less than 1% of the genome. So what we've been doing is also to complement this work with the GWAS studies. We've been doing large-scale genome sequencing for early onset atrial fibrillation. And I'll tell you a bit about, by the middle of next year, we'll have about 12,000 AF cases with full genome, high-quality genome coverage. And I'll talk a little bit more about those. So I mentioned that the slide that I showed you a moment ago was this one. In the next slide, we're going to look at these pulled together. And that's this. Now it looks less like a Manhattan plot and more like a forest. So this is work led by Carolina Roselli, an analyst in the group who joined us to kick off this project. This is PIDX2, which you'll now see is about the 1 times 10 to the minus 540 or something. It's always just going to get stronger as you increase sample size. But what the larger samples allow you to do is to look much deeper into the frequency of the variants that you can study, convincingly look at it. So the earlier slide that I showed you, the first studies that were done, we had to look at variants that were at a 5 to 10% frequency in the population. The one I showed you a moment ago was a 1% frequency of variants. This gets us down to 0.1. And when we do that, you come up with basically the top 100 loci when you use 65,000. So you quickly start to saturate. And that's why larger sample sizes here for genetics are always going to be more helpful. Now, the larger the sample size, the weaker the effect. So that will always be the case as well. If a genetic variant was going to have a strong effect, you would pick it up in a smaller number of samples. So what do these? It's a little hard to even process these things. I'm not going to put the names on the slide because they're kind of irrelevant. We did a bunch of downstream analysis. I'm only going to show you one because what you can see is if you do a gene enrichment type set, which I don't like most of these, I tried to break them down into you start to see 3 big themes occurring. And you start to get things involved in development, things involved in contractile function, and then a series of ion channels of related proteins. And this was, we manually curated this after we did these gene set enrichments, but that's the theme. I mean, just to try and make 100 loci something a bit more digestible. But as I was talking with some folks about earlier, only about, out of these 100, there are only 20 to 30 that you might say are really things that you could associate with atrial fibrillation. So the fun here is going to be to say, what do we do with all these things in the years to come? So, if you're a postdoc, you want to knock out one of these genes and study it, there's plenty to do, okay? I'm only going to be able to study one little bit of them. If you put everybody in this room together, you'd still only touch a fraction. I won't talk too much more about the individual level things. This is work that is under consideration at the moment, probably beyond a couple months. So one thing we're asked all the time, though, is what does it mean? What is the genetics? If you have this genetic fingerprint, who cares? Is it just a research tool, or is it something that could actually be of some downstream benefit? And then there was a neat little story. It's nice when stories are done by by the junior faculty members or trainees that work with me, and I'm just the bystander. So this is one of those. Steve Lubitz, who trained with me, has done exceptionally well, and his postdoc, Lu Chen, and then Sarah Price, who is based at Framingham, did a little project that I'm going to summarize in 2 slides. It doesn't really do it justice, but that's the way it goes. What she did was to take the genetic results that I showed you, the first one, where we had 24 genetic loci, and then they made a genetic risk score. So for the 24 variants, you get 1 copy from mom, 1 copy from dad. And so you can have, just in the simplest iteration, you can have 0 risk variants, or you can have 48 risk variants. And what they did is they then said, okay, well, that's the most stringent way to do it. Why don't we go lower and lower into the list? Okay? So instead of looking at the top 24, let's look at the top 50, 100, 1,000, 5,000. Let's go deeper and deeper into the list. And then also let's look around those variants. Okay, so look for weaker variants. And then let's look at all of the possibilities. So they looked at 30 possibilities. And the way they did this was by looking in the UK Biobank. So they basically optimized, looked at 30 different scores and optimized one that was, turns out that the best risk score for AFib consists of about 1,200 variants scattered throughout the genome. And then we applied it to Framingham to say, does that help us predict one's risk of atrial fibrillation? And there are 2 messages that came out of this. The first is that the lifetime risk of AFib is really high. So as we have an increasingly large, the obesity epidemic and other covariates associated with AFib, what we see is that if you're 55 years of age, right here, the chances of getting atrial fibrillation the rest of your life are 37%. It's huge, right? Now, mind you, this is an all European ancestry. Framingham is white. Anybody who was non-white in Framingham in the 1960s or 1940s was probably lost. And I don't mean that, it's just, you've got to accept it for what it is. This is the study design. But what we then did is say, okay, how does the genetics help us? Right? You can think of 2 big things to change. You can change clinical risk factors, and the risk of this should go up or down. And then if you change the genetic risk, what happens? And this is what happens. If you take a genetic risk score for AF, this 1,200, it actually allows you to stratify as much as a 20% gradient in the population. So this is this middle group here. We put them into high, intermediate, and low. And the intermediate should be right at the 37%, essentially this. But then if you have a high genetic risk, you have a nearly 50% risk of developing AFib. And if you have a low genetic risk, you drop down to 1 in 4. So you get this huge spread in the risk. And you can think of that a couple of different ways, and there's a lot more in this. I mean, this came out in sort of a couple weeks ago at the AHA. But what you can learn from it is that there's really a couple messages. If you can have a glass half full version or a glass half empty version, depending on whether you're the optimist or pessimist. So that if you're at a low clinical risk and you have a low genetic risk for AFib, your chances of getting AFib in the rest of your life are really, really low. But if you're a low clinical risk, so you're that athlete that comes in to see me all the time, and they say, I don't have anything wrong with me, doc, why do I have this? But if you have a high genetic risk, you have an exceptionally high risk of getting the disease. Now, if you look the opposite way and you take someone with all the risk factors and you say, why does this person that I see in the intensive care unit has every risk factor for AFib, and they don't get it, right? If they have that low genetic risk, they're pretty much protected. And then the opposite, if you put then a high clinical risk and a high genetic risk, you've got a much higher rate. So the optimist's way of looking at it is to say, if I have a high clinical risk and a high genetic risk, I can still reduce my risk by modification. So losing weight, controlling blood pressure, and still all the things a cardiologist will tell you. If you're a pessimist, you can say if you're that young healthy person, you have a high genetic risk. Sorry, buddy, training and all the exercise is not going to mitigate your risk dramatically. Anyway, that's just a couple of little vignettes out of the GWAS data. So I'm going to change gears a little bit and talk about some of the genome sequencing. The advantage of this GWAS type approach is that it's very cost efficient. That larger sample size where we genotyped about 35,000 individuals was $50 a sample. By contrast, genome sequencing is going to be in the range of $1,000 to $2,000. When we started, it was $2,000 a sample. Now it's down to about $1,000 a sample. And if it's cost efficient, you can get huge numbers, which increase your power. But we obviously have limited coverage of the genome. And the other is, if you're testing for a genetic variant, by default, you have to know what that genetic variant is. And if it's on one of these arrays, then it has to be a known variant. Okay? So it's not going to identify something like long QT syndrome, right? If you think about long QT syndrome, most of the genetic variants are de novo variants or unique variants that occur, and you find them by sequencing. So we're not going to get those. And now you can do genome sequencing is obviously expensive. Because it's expensive, you're going to have limited power. You obviously have the best coverage of the genome, aside from technical considerations. You have great coverage. It identifies known things, obviously, but it also identifies, as Rumsfeld would say, the unknown unknowns. It identifies the genetic variants that we don't know of and the associations that we don't know of. What we did for this particular study, this is Soo-Won Choi. Soo-Won's postdoc who joined the lab just over a year ago. We had taken 9 studies from the US and with early onset AFib, pooled them together and sequenced them. And the sequencing, I'm going to skip over almost all of the details. Happy to talk about any of those offline. But we had a great coverage, 99% coverage of the genome, 38-fold coverage. We identified 99 million variants, 8.4 of which were common. So we had really a high quality coverage across the genome. I'm going to skip all the analyses, but we started with 18,000 samples in this TopNet project. This was an NHLBI sponsored project. When you get to genome sequences that are a couple $1,000s a sample, you're not going to be able to run a perfect case and control study. No one's going to pay for that. So what you end up with is cases, and then you'll use all of the other samples and pull out a subset of controls. That's just a practical design consideration. So we did that. Most of these were non-white samples. All of our samples for this were of European ancestry intentionally, because that's where we have the best power. So we started with 18,000 samples, narrowed down the 9,500 that were of European ancestry. The Amish are a distinct subset. They were included in here. We dropped those, and then got down to 7,700 samples total. This is what they looked like. 2,800 with early onset AFib. So this is bigger than the GWAS studies we were publishing just 2 years, a couple of years ago. And we had 5,000 reference. There were obviously some differences in ages and covariates. We had selected these to be as healthy as we could, and there is some background rate of other things like diabetes, heart failure, myocardial infarction. I'll touch on them in a moment. But then the first analysis we did was really a quality control analysis. If you've got the whole genome sequence, you just look for common genetic variants, just like we did for GWAS, basically quality control. And you look at the Manhattan plot, and this is what we see. So because we have 3,000 samples instead of the 65,000 that I showed you a moment ago, we have reduced power. But nicely, what we see are things that we knew about. So the 24 loci that are published at the moment are here in blue, and all of them had an association in the right direction, although variable in strength. And mostly we saw what we knew. There was one other gene that kind of was significant. It's buried in that forest that I showed you. But this is a gene NAV2. It's actually, the cool thing about this one is that when you knock it out in a mouse, you don't get a vagus nerve. So when you think of young people with early onset AFib, often a vagal precipitant, and that's commonly talked about. It's kind of a neat association. That's necessarily causative, but it's an interesting association. But mostly we found what we expected. The next question is, what about those rare variants? Do you see an accumulation of rare variants in any other genes? So if you go back to the lung QT problem, if you sequenced 3,000 people with lung QT and 5,000 controls, you'd expect to find casing, you'd expect to find 1, 2, and 3, lung QT 1, 2, and 3 mutations in casein Q1 hERG in the sodium channel, and maybe a couple of other proteins as well. You should have pretty good coverage. And so that's what we did. One particular protein jumped out at us, and that was something well known, and that's titin. And what we found is that in people with early onset AFib, in the absence of heart failure, in the absence of any echocardiographic abnormalities that we could identify by echo, again, we don't have MRIs in all these people, but what we found was that this is just a cartoon of titin. It's the largest protein in our body, 120,000 amino acids, and goes from the Z disc to the M band. Basically, these are the different transcripts for titin here, in which each line is an exon. This is just a cartoon to show which part of the sarcomere it's in. And then up top here, each line, what we did in our analyses was to go to something that we thought would be fairly convincing, meaning that we just went for variants that truncate the protein. Okay? We're not looking for a SNP that slightly changes it from an alanine to a glycine or something. We just went for things that are terminations. It's stop loss or stop gain, essentially. Each line here is one of those variants. So you see about a doubling in the number in your early onset AF cases compared to your controls.
So they're not unique to EOAF and reference. They are amplified more in EOAF.
Each of these there were none that overlapped between the early onset AF and the reference. These were all unique events.
Is each one of them just a single mutation then?
Yes,
one
individual. Yeah, one individual, one loss of function. We had very carefully excluded any familial, cryptic familial relationship. That was all the details that I skipped, but we dropped all related individuals in this primary analysis. It's interesting, we can then go and look at transmission in a place like Framingham and see if you transmit, but this is an unrelated European ancestry analysis.
Just one question. So the reference, so those are the people who actually didn't have any early onset AF, and they still had mutations there.
Yeah. So it's a great question. Yeah, there are a lot of intriguing ways to play with this data. One is, we're pretty paranoid about it because, one is, what if it's just, it turns out that one of the sites, when you ask for samples, you set out some criteria. We set out very specific criteria. And then, sometimes when you say you're going to give people genome sequencing and it costs a couple $1,000s a sample, they happen to add in a bunch of their favorites. So, from the 9 different sites, we had a frequency of, 1.5%, 1.5%, 1.5%, 3%. It turns out that the group that was 3% said, oh, you wanted us to exclude the cardiomyopathies. Yeah, we forgot that. So we rediscovered titin for cardiomyopathies. So we dumped all of those. And then, of the 2,800 samples, we had a subset, because again, these are drawn from, like at Mass General, I've got lots of data. We put in about 1,000 of the samples. I've got tons of data on those people. But there's no indication to do an MRI on a 40-year-old with AFib, right? Well, this wasn't. We'll get there. But I think that's something that prospectively we'll start doing. But so what we did is, of the 2,800 people, we said, if anybody has any history of heart failure before or after the diagnosis of AFib, we're going to exclude them. And then if they don't have an echo, we're going to exclude them. And so that cost us about 800 people. So this plot is 2,000 of the cleanest early onset AFib that we could identify. There's still but it's a worry. The referent population, we don't know the AF status. So the reason is because of that pooled control problem, right? Which is it's too expensive. So in a subset of the referent population, a subset of this came from Framingham. There I know the AF status. I've got lifetime follow-up on the folks. But for some of the other studies, they just never checked. So this is basically, there's a background rate, and it has a few implications. When the heart failure folks look at titin mutations, they're actually saying it doesn't have any heart failure. But it turns out that in their referent population, they aren't excluding people with AFib. So my bet is that if you have a truncation in titin, most of these are probably going to have some manifestation of disease. I mean, this is going to be like the sodium channel. The sodium channel causes 6 different diseases. Titin is going to cause half those different diseases. Right? There's no way you're going to have the largest protein in your body and only have it cause one disease.
But you're not claiming it as a monogenic cause, right? No, you're just looking at it as a prevalence marker.
This is prevalence. Yeah.
So the reference could have it and still not have AF.
That's absolutely right. They could. I think that the closer and closer you look at this referent population, I think the more and more that the frequency will drop. Because if you start to exclude cardiomyopathies and or mild LV dilation or AFib, then what you'll see is the frequency will drop and drop. And that's what we've seen in some of the other control populations. We actually, in the next phase of this analysis, do have enough cases to actually just do AF, no AF. And we see that the background rate in the population is. So one other thing that we looked at was...
Sorry to interrupt.
No problem.
The expression of titin in the atrium and the ventricle.
Yeah.
Is that assumed to be the same?
The same? That's a great question. The folks who study heart failure have only really looked at its expression in the ventricle. We've done sequencing of about 100 normal people from the left atrium, and there are some interesting differences. We're still basically analyzing a lot of that data now. It looks like there are a number of exons that are basically dropped out, and then what we'll go back and look for is enrichment within those. Is there, what we'll see is there are some that are dramatically differentially expressed, some exons that are dramatically differentially expressed, and you would expect that those would be relatively enriched for AFib versus cardiomyopathies, which is something that we'll pursue. That's taking us a little longer to do.
So the reason I asked is because Hank Ranzier's work in it's in Arizona, showed that titin is a bidirectional spring.
Mm-hmm.
And that function of titin is substantially different in the ventricle and the atrium because of the macroscopic volume pumping.
Sure.
And so is there a way to relate these things to that kind of classical stuff?
I don't know right now. I mean, frankly, we I think you could look, Simon's had a nice story on looking at titin variants and on its contractile force measurements in iPS-derived cardiomyocytes. We could go and look for contractile versus a molecular effect. It could be that the atrium is like the canary in the coal mine. It's just more sensitive. And so we may pick up, we have data, we've got prospective data for some of these individuals. What I kind of assume that, maybe the AFib is just a predecessor to cardiomyopathy. To get AFib, and we see this all the time in other familial diseases. Dilated cardiomyopathy family or HCM family, pretty common. You'll have an index case over here, and their cousin will get AFib at an early age. Well, it's just basically you found HCM in that family. So I don't know. My sense at the moment from looking at the data is that there's probably an overlap. There are going to be some people with cleanly cardiomyopathy, some that just have isolated AFib, and you're going to have some in the middle where one or the other will precede each other. And that's pretty common with what we see clinically. When you look at DCM in general versus AFib, when you have an index event for AFib, the most likely next diagnosis you're going to have is not a stroke. It's actually heart failure. It's a 30% chance in 5 years. It's actually, and then vice versa. If you have heart failure, your next most likely major event is AF. So this is an intriguing molecular link, but I think there's going to be we've got a lot more work to do on this one. So, and the other thing is, if it's genetically driven, or we would expect the association to get stronger and stronger at an earlier and earlier age of onset. So what we see is that basically, although the confidence intervals are wide, for people under 30, which is a decent number of events, it's about 7% of the population. It's about 1 in 20 people. If you have AF in your 30s, they have a tightened loss of function variant. What that raises in my mind is, I still see people clinically, is that those 20-year-olds, which typically we talk about if they have paroxysmal and highly symptomatic AFib, we're talking about doing ablations. We're not necessarily talking about running a genetic test, which would cost $100 and we get back 6 weeks later, and we're not doing MRIs to look for subtle myopathies, but we'll start doing that prospectively. We've recently, as in this past week, what we did was to replicate this in the Regeneron data, so the Geisinger data. So for folks who aren't familiar with it, they have finished exome sequencing 92,000 exomes, including a subset with early onset AFib, and they saw this exact kind of increased frequency. So we're getting increasingly comfortable with this association. So it's a little bit on the common genetics and the genome sequencing. Questions at that point? Or I was going to tell you a little bit about one story. Sorry, Stacy, I did not get the chance to put in the mice, but I'm happy to talk to you about that afterwards. When you've got like 100 targets, we've made mice for some of them, but I'm also really intrigued about what causes some of these associations. Most GWAS loci are in non-coding regions, and they're presumed to regulate the expression of an adjacent gene. So they're not mutations in genes, that's pretty rare. Or a thing like Titan we wouldn't pick up with these GWAS techniques. But the one that's driven me a bit nuts is this top one, because we've known about it for a decade, and this is the unadjusted plot now. So instead of a Manhattan plot, this is a Dubai plot. This is one thing dominating everything else. And the question is, how do these variants lead to AFib? Now, as I said, when we started this, there was almost nothing known about it. But in elegant work by Vincent Christoffels and others, they've shown that PIDX2 is necessary for both left atrial and pulmonary venous development. Loss of PIDX2 by Brown and Martin and a number of other folks have shown that it leads to AF susceptibility in a mouse. So mice, which typically you wouldn't think of AFib, certainly do. And then just to emphasize that point, this was the first venogram from Michel Hausseger's paper on ablation of atrial AF, ablation of AF clinically, and the source coming from inside the pulmonary vein. This is a PIDX2, this is LAC-Z staining, PIDX2 in developing mouse. I mean, PIDX2 activation specifies this pulmonary vein to develop, and it's exactly where you get AFib coming from in a human. And it's a pretty complex little region. So if you zoom in now, this is a regional plot where you've gone from that. This is an older plot, so the numbers are a bit reduced, but this association is not in the gene. Okay, so the PIDX. So this is the minus log 10 in the p-value. This is chromosomal distance. Each dot is a SNP that we tested for association with AF, but PIDX2 is here. Then you've got this region here. This is driving AFib. Something in here is what's driving AFib. And you assume that there's an enhancer or repressor or something here altering the expression of PIDX2. It's kind of what we're assuming. Now, it doesn't have to be the gene next door, but I think given the other mouse work that's going on, we could feel pretty comfortable that the likely gene in this region is PIDX2. But the other interesting thing is if you take this region and then you adjust for this top variant will fall to the noise, and if there are other signals, they'll rise up out of this region. So there are now 6 different regions of susceptibility. There's one right over the gene itself. There's this top one, and there are a couple others here. Doesn't much matter, but the largest contribution to AF is driven by this one locus. And so there are presumably a series of enhancers or repressors at each of these different areas that are all interacting in tandem to alter the level, the transcriptional level of PIDX2.
I'm not sure whether you would, perhaps you would pick this up anyway, but is it possible that in that region where you have the high association, it's actually in linkage disequilibrium with a rare variant that's actually, say, in the PIDX2 locus, it's in the PIDX2 gene itself?
Yep. Yeah, it's distinctly possible. Turned out not to be the case for this one. What we did is, the first thing we did was to sequence the locus.
Okay.
And this little story was, now we started this about 5 years ago, so at the time it seemed like a Herculean effort. Now that we've just done genome sequencing on everyone, it sounds kind of passé. But what we did was we sequenced the PITX2 locus. We took 750,000 bases. We started just way up here and went way out here, and we sequenced it in 500 early onset AF cases, 500 controls, because we wanted to look for exactly that. Is there some variant here that's just an LD with something in PITX2? Maybe it's actually in the gene and we just missed it by a GWAS. It wasn't. And then the other possibilities, a couple other possibilities, there could be a structural rearrangement. Maybe just have some folks that have an inversion, a deletion. We didn't see any copy number variation. We didn't see any of those. So it's one of these common genetic variants in here that's driving the association. So then we wanted to, functionally assess this. This is Nate Tucker who did this work. We wanted to functionally test what about each of these individual genetic variants. How might they, if one of them is altering the function of an enhancer, how might they, or are they doing that? And so you could imagine there are only 200 variants in this little region. You could imagine just make the major allele, the minor allele, put up 200 luciferase assays and let your tech postdoc go to work. Grad students, I guess. They'll get back to you next year, and they'll do it. But and we've done that for, this story we published just a year ago, where we, did that and showed that we could disrupt one of these genetic variants, disrupted a transcription factor binding site that modulated the expression of PITX2. That took us 4 years to do that paper. This is like, these studies are, these functional genomic studies where you link a genetic variant to expression of a gene are tedious. And so we wanted to try and make a more high throughput way of doing this. I'll just describe that a bit. This is work we've just submitted. Just conceptually, since we know that the signal is here for AF, why don't we just test every variant at once? And the way that we did this is what's called a massively parallel reporter assay. So we took each variant, so say it's a C and a T. You can order all of those now just on plates, 6,000 at a time, or so you can have them synthesized on one plate. And so what we did was to synthesize 150 base pair oligos, where we put every genetic variant, the major and the minor allele, in either the middle, left, or right of a little region. And then we wanted to test and say, does this little fragment of DNA act as an enhancer? And if so, does this variant change that enhancer activity? Okay? So we just want to test them all at once. And now I'm going to focus in on the 262 SNPs here, but you can do this for thousands. And what we did was we put in not only the SNPs here, we put in everything across this region, positive or negative. So we put in a lot more. Okay? But you could do that. 262, like your grad student can manage that. What we fused is we then fused them to luciferase just as a dead reporter. If you can pick any gene. We put it upstream of a minimal promoter, and then we barcoded. We tagged on a barcode so we could identify. And this is done in a pool. Okay. So when you do that, what you get is then you just clone it into a vector. You can call it MPRA1. It doesn't matter. And then you take this pool, which now has many, many copies of each of these individual barcodes. So you're getting every SNP is represented multiple times in 6 different variations, and then it has many-fold representation in this plasmid library. And then you transfect that into your favorite cell line. Okay? And then
cell line? What cell line would you use?
We picked 3. So that's here. So at this one little locus, you're then, if you think about it, if you test 6 times the 262, you're at 1,500 combinations. But in reality, we're doing many, many ones. We did HL1, C2, C12, and HEC 293. There's no perfect. It's all an artificial system. But you can do this a lot of times. This is a trivial part. You transfect it, and then what you're doing is your readout here is it's a ratio of DNA, RNA to DNA is your enhancer activity. So you transfect in these plasmids, you sequence the DNA, and you're saying how many times was that plasmid there, and then you sequence the RNA, and you're saying how many times was this here. So just as a little cartoon model, this is a SNP with no activity. So you got a SNP here. This is, say one allele. This is the other allele. Doesn't make much RNA, so you see more DNA or an equivalent amount. No real activity. Other possibility is you could have an area that's very active, but but there may be no difference between the major and the minor allele. What we want to look for are things like this next guy, where you would see large expression on one allele and no expression or less expression on the other allele, and it's associated with disease, right? Because just because you test everything in the region, you actually may have some just by chance that are, have a differential effect. Maybe they have nothing to do with AFib. Maybe it's some other trait associated with PITX2 function, because it works everywhere and it works on a lot of other organs. So that's what we did. We come back to this focus. We then just plotted on this next plot. This is relative enhancer activity. And then this is this regional plot where I've just zoomed in on each variant that was tested. You get a few areas that drop out because they're highly repetitive. If you have a repetitive region, it doesn't make SNPs dropping out. And what you see are a lot of basal activity. There's some noise in the system. And then you see some things that jump out like this with, an 8-fold increased expression of RNA to DNA. And again, I'm skipping all the methods. I'm going to talk about them offline. The methods analysis was the longest part of all of this. The experiments are pretty trivial. And then, we thought, why don't we just look at the ENCODE databases? How does this line up to what we already know about? So you can turn on your favorite tracks. And some of them line up pretty well. So this is, a little area that's an enhancer. Here's skeletal muscle. It has enhancer activity in skeletal muscle. You get this one. That one lines up pretty well with skeletal muscle. And then you get something like this that doesn't really line up with anything. I think we have a limited amount of data for left atrial epigenetic data. So maybe it's just we have the wrong cell line, maybe we have the wrong time point. But what we really want is, this is any enhancer activity. What we want to then say is, how about that delta in activity, where one goes up and one goes down? And that's plotted on the next plot. And you see the same four SNPs kind of appear, slightly different, but this one where you have a strong differential activity. And then what we said is, okay, that's fine, but what about the association with disease? And that's this next plot. So you then take the minus log 10 of the p-value and plot it here, and you plot enhancer activity here. And you're going to see most have no enhancer activity or any association with AF. You'd hope so. Your guess is probably pretty lousy. Some will have a differential enhancer activity, but also no association with AF. You've got a bunch up here associated with AF, no enhancer activity or no differential enhancer activity. And then you see some things like this. And this was the core disease and differentially expressed. And then you can go back and validate that. Assay, you see a 10 versus 5-fold effect. The standard luciferase assay, we have lots of zebrafish in my lab. We don't use them as much anymore, but this is a zebrafish heart, where you see basically expression. This is transient expression of this enhancer. You see enhancer activity in the zebrafish. So we think this is a way. Now, it's pretty trivial. We were testing every variant. Pretty trivial for us now to do these types of assays, and that's what we're walking through and doing. It's just now that we have hundreds, some are not. We actually tried a couple in this. We had 2 loci that had a strong result, and one that didn't. It'll work for some things, it won't work for other. But what it allows us to do is do this very, very quickly. Okay? And I think that's the real power of this. That is for that functional variant identification, we can knock through many loci once. Tools for AF. So I've been, I'm one of the PIs on the Duke grant with Vincent Christoffels, Dr. Demat, Paulus Kirchhoff, Jim Martin, and Ivan Moskowitz. It's been a great collaboration. I learned a lot from these guys, and the things that we're doing at the moment are working on basically trying to build out a better epigenetic tool set for the left atrial tissue from our human samples. And any questions about this bit? Just leave you with one last thought on the therapeutic targeting. I don't have any advanced data to show you quite yet. As I said, it's only been going on 2 years, but what's the rationale for it? This is the best clinical trial of our antiarrhythmics from Bram Singh back in 2005. Randomized controlled trial looking at amiodarone versus sotalol versus placebo. And a couple of observations for this. If you took your kids in for a strep throat and you gave them, and they got a drug that was like sotalol with an 80% chance of failure, you would think the person's a quack. And yet these are the two best drugs that we have. Even amiodarone, which works. You know, for some people it's like magic and it works. And if you're an 85-year-old and you have AFib and amiodarone works and you don't have toxicity, it's a great option. It's cheap, you know, you can get monitored. If you're a 40-year-old, this is not a drug for you. And Sonolol has real toxicity, and obviously, at even just a 3-year rate, you have a chance of success of only 25%. And this was before we had very intensive monitoring, right? These are clinical recurrences and trial-based recurrences. So there's a lot of room. I mean, this is why people go for ablations, because the line for ablation is up here, right? I mean, there may be drop-off, but you know, in most labs, most good academic labs, you've got a 3-year rate of 80+% for paroxysmal AF. You know, you're the low end. For persistent, you're probably down at 60 or so. But it's still beating these drugs. So the concept behind this collaboration, which is effort that I lead between the Broad and Bayer, I did on with my colleague State Catherison, who has done all the work that we've done for AFib genetics. He's done for myocardial infarction and lipids, just better. So we've taken these genetic targets and basically said, can we identify individual potential targets? You know, conceptually, this is the SK3 channel, or KCNN3, which was one of the early, identified that associated with early onset AFib back in 2010. And then can we do high throughput screens? I don't do this in my lab. This is some of the robots at Bayer. And then can we identify, this is not a new SK blocker, sorry. This is, we don't have that quite yet. But we've done this for SK3. We've made a high throughput assay. We've in cell lines. We've screened the combination of Bayer's library plus the Broad library. That was 4.2 million compounds with a 15-day runtime. And we had 6,000 primary hits, 2,500 of which you know, we filtered out, and we're now working on representative classes, target classes, to refine some of the structures. And it may or may not work. I mean, frankly, you know, drug developments, you know, there are a million reasons. It may go into the brain and cause someone to have a seizure. It may not be absorbed. It may cause you to drop over dead. You know, like, I don't know. Like, you know, there are a million reasons drug development can fail, but at least it's fun conceptually to think about. So with that, that's ongoing work. I'll just leave you with a few thoughts. You know, within the next year, we can anticipate 100+ loci for AF. Titin and loss of function variants are found in individuals with early onset AFib. Both of those stories I expect we'll see out in the next 6 months. You know, genetic risk can help you refine the lifetime risk of AFib. And then genetics, we hope, provides a window into therapeutics, but we'll see. I think stay tuned for that. And then one last thing I wanted to leave you with is that, you know, I think I've come to appreciate that this genetic data can be a bit daunting to navigate for a lot of folks. So one thing that we've really wanted to do, and that my colleagues at the Broad, I believe in, is trying to get this out into the public sphere and to make it user-friendly as quickly as we possibly can. So at AHA, we released a website to try and make this a lot easier to navigate. That's here. This is, we call it Knowledge Portal. It isn't actually a website, where what we've done is to take all of the genetic data that we've ever published for AFib, and it's all here, and it's all searchable. So, let's see. The Wi-Fi is working. I'll show you. It's easier to see. Take PIDX2. What it will do is summarize the data. So it gives you a little stock bite. You know, it's green, it's good. So then the phenotypes are here: AFib, P-wave terminal force, P-wave duration, and then it'll show you any other additional phenotypes. We don't have everything in the human body there. We have the people who are nice enough to give us the data, which mainly means the other people sitting around me at the Broad who I said, give us all your data. And you know, these are some of the top level phenotypes. We'll be adding probably 50 more GWAS data. So that what we've done is we've taken all the GWAS data for these different traits, integrated them, and then made the cross lookups that are already done for you. Okay? So what it will do is default to something with the strongest association. So this will then give you the top variants for AFib. You can then look and say, you had asked earlier about, are there any coding variants that are there? You can flip to that and see if there are. And then with the common variants, it'll also just show you the regional plot. So this is now the regional plot for AFib. If you want, you can scroll it over in real time. It'll recalculate it for you. If you press on an individual variant, it'll tell you anything you want. We've loaded in we'll load in all of the epigenetic data, but if you want to see, we can split that out, and you can look at individual enhancers that might be in the region, and then we'll keep adding to this. The other data sets that are in here so we've tried to make this a bit more user-friendly. These are the traits that are in there right now. So coronary disease, AF, lipids, ECG parameters, we'll add a bunch, diabetes, and psychiatric disease, renal disease. So anyway, if you have a favorite gene, feel free to punch it in. We'll give you everything that's published to date. So with that, I would say that I give a lot of PowerPoint talks and write a lot of grants. The people in my lab that actually do all this hard work, I've tried to highlight as we go along. But I have a great group of people that work with me, and the folks who led most of these studies. Sung-Won did the genome sequencing, Nick Tucker the NPRA, Carolyn did the latest genetic analyses. So thanks very much, and happy to
Question to Patrick.
Please.
With the growing understanding non-coding RNA or non-coding region with your genetics from there, do you think that's maybe an alternative strategy compared to altering the transcription activities of transcription factor using microRNA or other RNAs?
There's certainly some, links and some microRNAs at some of the loci we've identified. Some of the loci it's pretty obvious, or it seems pretty obvious. The challenge is with the GWAS data in particular. The challenge is that you might get a genetic locus where there's one really obvious thing, and the genetic variant sits right on top of it. So for SK, it's SK3. So that's a good example. Like PIDX2, we have pretty good data, but in a different gene, CAN2, it sits smack in the middle of the gene. There's a strong EQTL. So we'll often look and say, does a genetic variant link to a gene in that region? Meaning, does the genetic variant link to expression of the gene? And if you see that, a strong association between that variant and expression of the gene, then that's probably your gene. But a lot of loci are not so obvious. You might have 10 different things. And within those are a bunch of links and microRNAs. So I'm sure that some of the loci will probably trace back to those. Most of our thinking and most of the studies that are designed to look at this are really targeted at genes at the moment. As I said, there's tons of work to do. And you could pick a favorite locus, you could pick a favorite gene, there'd be many years worth of work to do. And I welcome anyone to play with these data sets. We're happy to. If you want the whole dense data set, I'm happy just to email you a copy of that. Well, I'll give you the link and you can download it. It's a little more than an email. But you're welcome to play with the bigger data set. Yeah.
So for current clinical practice, so every once in a while I'll see a patient like 25, 30 years old with AFib. So I'm not going to do a whole genome sequence on them right now. So what would be your recommendation at this time? What's ready for prime time, I guess?
So at the moment, I wouldn't even necessarily do panel testing on them. I think our obligation, this is the first result. It's kind of our obligation to then say prospectively that this is meaningful. So we're doing those types of studies now, where we're taking every young person that comes through with AFib. We will run a standard genetic testing for clinical purposes. Well, we're doing a research background, but with a genetics counselor if they have any alteration or loss of function variant. But I think we have to look at that independently, completely independent from this research study. We have to look at it prospectively and reconfirm it. I think that that's kind of the next step. So I wouldn't go altering clinical practice based on what we've seen so far. I think we have to do a completely independent replication of this work, which I don't think will take long. I mean, we see a lot of young people with AFib.
So the GWAS consortia, they persistent, paroxysmal, lone AFib, all of the above, and I guess where I'm going with it, are there specific variations that lead to persistent AFib, some that will just be sort of transient, and never lead to persistent? I mean, what kind of
phenotypic resolution do we have? Yeah, it's a great question. When we first what I showed you there is everybody. You can be a lumper or a splitter. This is everyone. and you do that to get better power. Obviously, when you do that, you give up the resolution. So it depends a bit on the study. The very first 2 genetic studies that we did, we had exactly your concern. And we took 5 studies with early onset AFib, because we just said, like the young people that are 4 years old and have AFib are totally different clinically than that person in the ICU. So we actually ran them independently. We took 5 studies with early onset AF and analyzed those. We took 5 studies with an average age of 75, so Framingham, Eric, CHS, Rotterdam, and ages and there the mean age was 75. What was kind of surprising is that they almost overweight perfectly. And there were differences. There were differences, but there were more similarities than there were differences. So PIDX2, strongest thing in both of them. The second genes, the next one to come up in young people was KCNN3 or SK3. The next thing to come up in older people was a different transcription factor. But they were all there. Now, the younger you are, the stronger the genetic basis. So it's a much, much stronger effect. So PIDX2 in that 75-year-old in the intensive care unit would have a relative risk of about 1.5 to 1.6, so a 50 to 60% increase. In a young person, it's about 2.5-fold, so about a 250% increased risk. So, really striking at the young ages, but mitigated. So it's an age-dependent. Does that make sense? What we're doing now, to get at your question, is I've been reluctant to subdivide our population when it was small, because what you want to do with these genetic studies is just bring overwhelming force to them.
Right.
And so when we've done studies, that's why we've lumped everyone together. When we had 1,000 people, it didn't make sense to really divide them up into paroxysmal. There are other really cool questions of like who gets heart failure, who doesn't, who gets a stroke, who doesn't. Now when you're at 65,000, we can still be well powered to do those sub-studies, so we're doing it now. Yeah, that's ongoing.
With those associated SK3 variants, it's pretty easy to test the functional consequences. So are they all gain of function?
They're not in the gene itself. I mean, those SK3 variants are not in the gene.
Do they cause increased expression? Did we learn that?
We have just basically seen that.
Because if you're making a blocker, you'd better be right that they're a gain of function, right?
Well, when you do the screen, we get both blockers and we get both agonists and antagonists. It was a bidirectional screen, but you're right. You're not going to take it into people until you're sure of the direction of the effect. Mm-hmm. There are other non-specific blockers of the SK channels, like Arrhythmia, the bee venom toxin, that causes action potential propagation. But what we're looking for is a more specific blocker of SK3 and or 2. So what was also interesting in the genetics for the SK channels is that, like other potassium channels, they're multimers, and they're typically heteromultimers. So in the atrium, there's expression of 1, 2, and 3. But what's kind of cool about it is that SK3 came up very strongly in the latest work we did, so SK2. So you're now seeing 2, and they're completely different loci. You're seeing 2 of these constituent members of this genome complex that come up independently. So we're hopeful.
So with the drug development, the idea of the rationale for developing drug, I just want to ask you. So now you show the Titan and also PIDX2, and those change the structure of the tissue, right? So mostly, but AF is mostly electrical problem.
Maybe.
No, I mean eventually the presentation is symmetrical, right?
Well, maybe. I mean, maybe it's I think what you see with a lot of this is that a big number of the genes that we've identified are actually like members of the sarcomere, or interacting proteins in the sarcomere. So again, maybe it's just that there's an electrical manifestation of an underlying myopathy, or some abnormality.
Yeah. So the underlying reason can be structural. So in the end, it is the electrical activity is affected.
Yeah.
So how do you develop the drug? Do you use the target? Do you use those structure gene as target to drug, and how do you work?
Yeah, we have not. We've not taken on things that are direct. You know, like I've not tried to find a Titan modifier,
Yeah,
for example.
right.
But there are, you know, there's some really cool mechanisms of regulation, indirect mechanisms of regulation of these proteins. So we've seen a couple of, so the story that I was going to tell that I didn't, you know, pull together all the slides for today. You know, one of the genes we were really captivated by was this gene called CAN2. You know, there are some total of 2 publications on it, which shows it's a member of the ubiquitination complex and it regulates myogen and skeletal muscle. The number of papers in the heart are 0. It has expression just in the heart and the skeletal muscle. And it had a strong EQTL. The genetic variant sits in the gene, it regulates the expression, so, and it's, looks like, you know, perfect thing. You know, we then said, we've done a bunch of studies on it over the last 2 years. We pulled out everything that interacts with CAN2, and you basically get the entire sarcomere. And then, because it's a member of the ubiquitination complex, we actually did studies to look at everything differentially ubiquitinated when you either have or lose CAN2. And it was essentially, again, all the targets were larger sarcomere proteins. So there are methods of regulation of sarcomere stability, be it ubiquitination or others, that I think are probably an entryway into that myocardial function. But these are early days. I mean, you know, that's a lot of fun work in the lab at the moment. There are 2 separate regulatory mechanisms which we're studying of contractile function related to AF, but give it another year or 2, and I'll have some better stories for you. Yeah, Stacey. Yeah.
So, in going from the GWAS to the mechanism,
Yeah.
Being that many of the enhancers are very cell type specific, even LA versus RA, you know, using cell culture models versus in vivo, how many of them are conserved at a sequence conservation level between organisms, for example? And also, you know, your choice of cell lines, what advice do you have along that line?
We need better tools.
Right.
So a lot of these things are not conserved. I mean, we've got a couple of examples, one at the PIDX2 locus, one at the PRX locus. The actual, the region that these variants were in tends to be conserved, but the actual base pairs that are, you know, denoting the site tend not to be, or, you know, may not be conserved. So that's a limitation. So there would be species specificity. I mean, you would hope there's some difference between us and mouse, but that's certainly a limitation. With respect to the cell line tools, I mean, you know, the HL1s look more like a right. You know, we've sequenced them; they look more like a right atrial cell line than a left atrial cell line. You know, we don't have a perfect representative model. You could consider doing these things in primary tissue. That would be hard to do at scale, the scale we're talking about. You could, instead of transfecting them, just virally deliver these types of things. I just don't know that you'd ever have enough human primary tissue. You could look at doing it in an IPS cell, but as we talked about, the IPS cells are a long way from a human adult atrial as well. So at the moment, I think it'll work for some things, it won't work for others. I think even if it gets you 30%, 20% of the well side, it's a whole lot faster than that 3 to 4 year story that we've spent on other well side. Because I mean, you know, now that we've got the methods for doing these parallel reporter assays, it's pretty trivial. I mean, you know, we're talking like a month. We get all the results back. You know, so it's basically a sequencing experiment. We have plenty of sequencers. So that's really one. So I think it'll, you know, be helpful at some of them, and for others, you're still stuck.
So if I understand it correctly, there is no clustering in the association studies between the young group and the over 75 older group, right?
No, there was a lot of overlap between them.
There was a lot.
And they almost superimposed on each other, although there was a stronger association in younger people.
For the genetic basis of in the association.
Yes.
But that doesn't mean that the target will have the same effect in the young group and the old group.
Nope. Yeah, we don't know.
I mean, you can develop whatever you want to develop, but that will be modified by the substrate.
Sure.
And there is also the delivery of the drug and the distribution of the drug in the tissue.
Absolutely.
Because the substrate is hugely different between the old and the young, hugely different. I mean,
Yep.
there's all studies from Medispa that for example, the degree of anisotropy is much higher in the old heart, especially in the atrium. And also that there is, of course, a lot of fibrosis and connective tissues like that. That will affect any interaction with the substrate. So the question is how, I'm just looking for the rationale of how we
So, you know, you can probably tell I'm fairly cynical at baseline, and I'm not convinced any of it will work. I mean, I think we have to try it. I think you have to be realistic about it. You know, like, why do we think we're going to develop an antiarrhythmic when it hasn't been developed by pharma in, you know, all these years? You know, I think one is when you've got the genetic data, you push a lot harder on targets than you would otherwise. So, you know, I've seen that in Bayer, I've seen it in other companies. When we say SK3 is a potential target, you know, we won't just stop at one thing. We'll come at it with many different approaches. That's how you got a PCSK9. You know, you saw this whopping association in humans. There's still no orally available PCSK9 inhibitor. I mean, it's all antibody mediated. But what it gives you is confidence to push on. Will it work? I don't know. I mean, I think to be realistic about it, you know, drug development is, you know, most scientists and drug companies are lucky if they ever get one drug in their life. You know, that would be a home run. For me, academically, we'll have a lot of fun and we're going to learn an awful lot along the way. So even if that's all we do, I think it'll be worth it. Yeah, but we'll have to see.
All right. Any other?
Patrick had a question.
I'm sorry.
Thank you very much again.
Patrick?
Do you see nonlinear genetic interactions between loci on
I'm
risk?
sorry?
Do you see nonlinear interactions on risk?
Not really. I mean, we've tested. We've looked. So genetic interaction studies require massive numbers for environmental or gene interaction studies. And we've actually done it for just the top loci, and we don't really see much. I mean, there was one report from a Chinese group on an interaction between PDX and ZPHX3. We were not able to replicate any of that. So far, we've really seen additive effects and not, you know, as you've mentioned, non-linear effects. All right, thanks very much, everyone.